Welcome to the deep dive. It's kind of fascinating. When you think about it, your DNA, it has to be incredibly stable, right? Like rock solid to hold all that information.
Absolutely. It's the blueprint.
But then here's a twist. Life itself, evolution, adaptation, it all depends on that blueprint changing. These little shifts, mutations, they're actually crucial.
That's the core paradox, isn't it? This balance between needing DNA to be super reliable, but also needing it to, change sometimes for life to move forward.
And that's exactly what we're diving into today. Here at the Deep Dive, our whole mission is to take complex stuff, like from sources such as Robert Brooker's genetics, analysis and principles, and really pull out the key insights for you.
We want to make sense of it all. So today, we're exploring gene mutation, DNA repair, and this thing called recombination. It's all about how our genes change and how our cells deal with it.
So why should you, listening right now, care about this molecular deep dive?
Well, because these processes are fundamental. Mutations drive evolution, create diversity, think different eye colors, everything. But they're also behind many inherited diseases and things like cancer. So understanding this really gives you a lens on life's mechanics.
Okay, let's get into it then. Mutation. What's the core idea we need to grasp?
The key thing, I think, is that a mutation isn't just a typo. It's a heritable change in the genetic material. Heritable is the crucial word.
Meaning it can be passed on.
Exactly. Passed from a cell to its descendants, or if it happens in sperm or egg cells, passed from parent to child. This is like the ultimate source of all the different versions of genes, the alleles that make populations diverse. Tall P's, dwarf P's, it starts here.
Right. And we often think mutation means bad, but you're saying it's not always negative.
Not at all. It's a whole spectrum. Some mutations are beneficial, tiny advantages that evolution can grab onto. Many are just neutral, have no real effect. But yeah, statistically, a random change is more likely to mess something up than improve it.
So let's zoom in. What do these changes actually look like at the DNA level?
Okay, so at the molecular level, the simplest is a point mutation. Just one single DNA base pair gets swapped for another.
Like changing one letter in a long word.
Pretty much. And there are two main flavors. A transition is swapping like a purine for another purine, say A for G, or a pyrimidine for another pyrimidine, C for T. A transversion is swapping categories purine for pyrimidine, or vice versa.
Okay, swaps. What else?
You can also have dilutions or additions where a few base pairs are either cut out or squeezed in.
And how do these tiny changes ripple outwards to affect, say, the proteins that DNA codes for?
Well, that depends. Sometimes a silent mutation happens, you change a base, but because the genetic code has some redundancy, multiple codes for the same amino acid, the protein actually stays the same. No harm done.
Lucky break.
Yeah. But then you have missense mutations. Here, the base change does swap one amino acid for another. Sometimes it doesn't matter much if the new amino acid is chemically similar, but sometimes it's huge.
Like sickle cell.
Exactly the classic example. One single base change in the beta-globin gene swaps glutamic acid for valine, just one amino acid difference. But it makes the hemoglobin protein wonky, causing red blood cells to warp into that sickle shape, especially when oxygen is low.
And that leads to all the health problems. Blockages, pain.
Organ damage, anemia, a massive impact from one tiny change.
Wow. Okay. What else can go wrong?
Well, you can get a nonsense mutation. This changes an amino acid codon into a stop signal, like putting a full stop right in the middle of a sentence.
So the protein just stops being made halfway through.
Yep. Usually results in a short, useless protein fragment. And maybe the most deceptive are frameshift mutations, if you add or delete bases, but not in a multiple of 3.
Oh, right, because the code is read in 3s.
Exactly. You shift the whole reading frame downstream. It's like deleting one letter in a sentence, and suddenly all the following words are just complete gibberish. The resulting protein is almost always totally non-functional.
We always focus on the protein coding bits, the genes themselves, but DNA has huge stretches that don't code for protein. Can mutations there cause problems too?
Oh, absolutely. Those non-coding regions are packed with control switches. A mutation in a promoter, the on switch for a gene, could make it hyperactive or shut it down.
So too much or too little protein?
Right. Or mutations in regulatory elements or operator sites can mess up how the gene responds to signals. Even changes in the untranslated regions, the UTRs at the start or end of an mRNA molecule, can affect how stable the message is or how well it gets translated.
And splicing. I remember that being important.
Crucial. Mutations in the splice recognition sequences mean the pre-mRNA molecule doesn't get edited correctly. Introns might be left in or exons skipped.
Yeah.
leads to faulty proteins.
So besides the molecular details, how do biologists talk about mutations based on what they do to the organism?
We often classify them by their effect on the phenotype, the observable traits. We start with the wild type, which is just the most common version in a population. Anything different is a mutant allele. Sometimes, a reverse mutation can actually change a mutant back to the wild type. Many mutations are neutral. They don't really help or hurt. Then you have deleterious mutations, which reduce survival or reproduction.
And the really bad ones.
Lethal mutations, which cause death. But on the flip side, though much rarer, are beneficial mutations, the ones that give an advantage. These are the fuel for natural selection.
And conditional ones, you mentioned those.
Right. Conditional mutants, their effect only shows up under certain conditions. A classic example is temperature-sensitive mutants, maybe in bacteria, that only show a defect when it gets too hot or too cold.
Okay, now what about these suppressor mutations? They sound like a kind of genetic backup plan.
They kind of are. Suppressor mutation is interesting. It's a second mutation at a different spot in the DNA that cancels out the effect of the first mutation.
So it doesn't fix the original mistake, but it compensates for it somehow.
Exactly. If the second mutation is in the same gene as the first, it's intragenic. Maybe the first mutation made the protein fold wrong, and the second one helps it fold correctly again, even with the first mistake still there.
Huh. Clever.
And if the second mutation is in a different gene, it's intergenic. This often happens when proteins work together in a pathway. A problem in one protein might be compensated for by a change in its partner protein.
This is getting wild. So even if a gene sequence is perfect, just moving it around on the chromosome can mess things up.
Absolutely. That's called a position effect. It's not about the gene's code, but its neighborhood. Large chromosomal changes like inversions or translocations can move genes.
And the neighborhood matters. How?
Two main ways. One, the gene gets plunked down next to some powerful regulatory sequences belonging to another gene, maybe an enhancer that ramps up its activity unexpectedly.
Correct.
Two, the gene gets moved from a region of active DNA called euchromatin into a tightly packed, silenced region called heterochromatin. It's like moving an active office into a locked vault. The gene basically gets shut down.
Is there a good example of that?
Yeah, in Drosophila, the fruit fly, sometimes a gene-controlling eye color gets moved near heterochromatin. The result? A variegated eye patches of normal red cells and patches of white cells where the gene got silenced during development really shows how location impacts expression.
That brings up another key point. Timing. Does when a mutation occurs make a difference?
Huge difference. This comes down to germline versus somatic mutations.
Or germline. Those are the sperm and egg cells.
Or the cells that produce sperm and eggs, yeah. If a mutation happens in the germline, and that gamete is used for fertilization, then literally every single cell in the offspring will carry that mutation. And crucially, it can be passed to their offspring.
So that's how inherited diseases get passed down.
Precisely. Now, somatic mutations happen in regular body cells, skin, muscle, liver, whatever, not the germline.
So they aren't passed to kids.
Correct. The consequence is within that individual only. They become a genetic mosaic, meaning some parts of their body have the mutation and others don't.
How much of the body is affected?
Depends entirely on when it happened. A mutation early in embryonic development, you could have a large patch of affected tissue later in life, maybe just a small, localized group of cells. Like someone might have a patch of white hair because of a somatic mutation in a pigment cell lineage that happened early on.
Right, okay. This really highlights why things like, say, X-rays during pregnancy are avoided protecting the developing embryo from somatic mutations.
Exactly. The timing is critical.
Now, there was this whole historical debate, wasn't there, about whether mutations just pop up randomly or if the environment somehow directs them.
Oh yeah, the Lamarck versus Darwin debate, essentially. Lamarck thought organisms could acquire traits based on need and pass them on. The modern view, random mutation theory, says variation happens by chance and then the environment selects.
So how do they settle that? The Liederberg's experiment, right? That's a famous one.
It's a classic. Joshua and Esther Liederberg in the 50s. Super clever setup. They grew bacteria, E. coli, on a regular petri dish, the master plate. No stress, just food. Then they took a piece of sterile velvet, pressed it onto the master plate to pick up cells from all the colonies, and then stamped that velvet onto several new plates. These new plates contained a virus, phage T1, that kills normal E. coli.
Ah. So only resistant bacteria would survive on the new plates.
Exactly. Now, the question was, Did the resistance mutations happen after exposure to the virus, or were they already there randomly on the master plate?
How could they tell?
If the mutations happen randomly before the virus exposure, then the resistant colonies on all the replica plates should be in the exact same spots because they're just copies from the master plate. And that's precisely what they saw. The resistant colonies popped up in identical patterns on all the phage plates. It proved the mutations were already present by chance on the original plate. The virus just selected them.
So random variation first, selection second. Case closed.
Pretty much solidified the random mutation theory, yeah. Though, we now understand rates can vary a bit. Some genes or spots might be slightly more prone to mutation, but the underlying event is considered random regarding adaptation.
Okay, so mutations can happen spontaneously. But what about things in our environment that cause them? Mutagens.
Right, induced mutations. These are caused by environmental agents called mutagens. They directly damage DNA and crank up the mutation rate way above the spontaneous level. This is why they're linked to cancer and why we worry about them.
What kinds are there?
Broadly, chemical and physical. Chemical mutagens work in different ways. Some directly modify the DNA bases, chemically altering them so they don't pair correctly anymore. Nitrous acid is one example. Others, called alkylating agents, stick chemical groups onto the bases, messing up pairing.
Nasty stuff.
Then there are intercalating agents. These are flat molecules that literally slide between the DNA base pairs like shoving a card into a deck. This distorts the helix and tends to cause insertions or deletions during replication frame shifts. And finally, base analogs. These are chemicals that look very similar to normal DNA bases. The cell gets fooled and puts them into the DNA during replication. But these fakes often pair up incorrectly later on.
Is that how some chemotherapy drugs work, like 5-bromoresol?
Exactly. 5-BU looks like thymine gets put into DNA, but it can sometimes flip its chemical state and pair with guanine instead of adenine. This causes mutations, which preferentially kills rapidly dividing cancer cells. But of course, it also hits other dividing cells, hence the side effects.
Right, and physical mutagens, radiation.
Yep. Ionizing radiation X-rays, gamma rays is high energy. It penetrates deep and creates damaging free radicals. These can cause base deletions, break one or both DNA strands, create crosslinks. Really messy.
And the sun, UV light.
That's non-ionizing radiation. Less energy, doesn't penetrate as deeply, mostly affects skin. Its main damage is creating thymine dimers. It covalently links two adjacent thymine bases together.
Like welding them.
Sort of. And that bulge really messes up DNA replication and reading, leading to mutations. That's the direct link between sun exposure and skin cancer risk. Sunscreen works by absorbing or blocking that UV light.
So we talk about mutations happening.
Yeah.
Is there a way to measure how often?
Yeah, we distinguish between mutation rate and mutation frequency. The rate is the chance a specific gene will mutate in a single generation or cell division. It's usually very low, maybe 1 in 100,000 to 1 in a billion. Fun fact, you probably have 100, 200 new mutations your parents didn't have.
Whoa. And frequency.
Frequency is just how common a mutant gene is in a whole population. It's a snapshot, not about new events.
How do scientists even test if some new chemical is a mutagen? Is there a standard way?
There is. The Ames test, developed by Bruce Ames, is the gold standard. It's really clever. It uses special strains of Salmonella bacteria that have a mutation, making them unable to make histidine, an amino acid they need to grow.
So they can't grow unless they get histidine.
Right, unless a new mutation happens that reverses the original defect of reversion. If that happens, they can make histidine again and grow.
Okay, so how does the test work?
You take the chemical you suspect is a mutagen, mix it with these special Salmonella, and, crucially, add a bit of rat liver extract.
Liver extract, why?
Because many chemicals aren't mutagenic themselves, but our liver enzymes can metabolize them into mutagenic forms. The extract mimics that. Then you spread this mix on a petri dish that lacks histidine.
Ah, so only bacteria that have undergone that reverse mutation will be able to grow and form colonies.
Exactly. If your chemical causes significantly more colonies to grow compared to a control plate without the chemical, bingo, it's mutagenic. It works really well. Testing urine from smokers shows way more revertant colonies, for example.
It sounds like our DNA is constantly under attack, spontaneously and from the environment. It's amazing we're not just riddled with errors. Our cells must have defenses, right?
Oh, absolutely critical defenses. DNA repair systems are constantly working. Without them, the mutation load would be completely overwhelming. Think about inherited diseases like xeroderma pigmentosum, caused by faulty repair. Those individuals have extreme sensitivity to sunlight and massively increased skin cancer risk because they can't fix UV damage properly.
So how do these repair systems generally work?
Most follow a basic pattern. Step 1, detect the damage. Step 2, remove the damaged part. Step 3, use the undamaged opposite strand as a template to synthesize the correct sequence and fill the gap.
Makes sense. Are there different types of repair?
Loads. There's direct repair, where an enzyme literally just reverses the damage. Like, an enzyme called photolyase uses light energy to break thymine dimers caused by UV. Simple and clean.
Like hitting undo.
Kind of. Then there's base excision repair, or BER. This tackles small problems, like a single damaged or incorrect base. An enzyme called DNA N-glycosylase recognizes and snips out just the bad base. Then other enzymes cut the backbone, a polymerase fills the gap, and LIGA seals it.
Okay, for small stuff, what about bigger problems?
For bulkier damage that distorts the helix, like those thymine dimers again, or large chemical additions, there's nucleotide excision repair, NER. This system recognizes the distortion, makes cuts on both sides of the damage, removes a whole chunk of the strand containing the lesion, and then polymerase and ligase rebuild it.
And defects in NER cause xeroderma pigmentosum.
Yes, and Cockayne syndrome, too, really highlights how vital NER is, especially for dealing with UV damage.
What about mistakes made during DNA copying itself? Does the cell double-check?
It does. That's the job of the mismatch repair system. It catches errors that the DNA polymerase missed during replication, like putting an A opposite a G.
How does it know which base is the wrong one, the original or the new one?
Great question. It uses a clever trick, at least in bacteria. For a short time after replication, the old parental DNA strand is marked with chemical tags, methylation, but the new strand isn't tagged yet. The mismatch repair system recognizes this difference and knows to fix the error on the new untagged strand.
Smart. And human defects in this cause problems too.
Yes, defects in human mismatch repair genes are strongly linked to certain types of inherited colorectal cancer. Shows its importance in preventing mutation accumulation.
Okay, what about the really catastrophic damage when both strands of the DNA helix break?
Double-strand breaks, DSBs. Yeah, those are bad news. Can easily lead to chromosome rearrangements or loss of genetic information. Cells have two main strategies here.
Which are?
First, homologous recombination repair, or HRR. This is the high-fidelity option. It uses the identical sequence on the sister chromatid, the duplicated chromosome copy, as a perfect template to accurately repair the break.
Error-free. Sounds good. What's the catch?
The catch is you need that sister chromatid nearby, so HRR mainly works during and shortly after DNA replication, S and G2 phases of the cell cycle.
So what happens the rest of the time?
The cell uses non-homologous end joining, or NHEJ. This is faster, more like an emergency patch job. It basically just grabs the two broken ends and sticks them back together.
Doesn't sound very precise.
It's often not. NHEJ frequently introduces small insertions or deletions at the break site. It's error-prone, but it prevents the chromosome from completely falling apart, which might be worse. So it's a trade-off.
And sometimes the main polymerase just gets stuck at damage.
Right. Sometimes the damage is too much for the high-fidelity polymerases. That's when translesion DNA polymerases, or T-less polymerases, come in. These are like the off-road vehicles of DNA synthesis.
How so?
They have a looser, more accommodating active site. They can actually replicate over damaged DNA that would stop the main polymerases cold.
There's a cost, right?
Big cost. Low fidelity. Because they're less precise, they make more mistakes, they're error-prone. But again, the cell sometimes decides that getting replication done, even with a few errors, is better than halting it completely.
Okay, so repair is all about fixing mistakes. But you also mentioned recombination. That sounds more like rearranging things intentionally.
That's a good way to put it. Homologous recombination isn't just for repair, it's also a fundamental process for shuffling genetic information. It involves the physical exchange of DNA segments between similar or identical molecules.
And this happens during meiosis, making sperm and eggs.
Exactly. Crossing over between homologous chromosomes during meiosis is a form of homologous recombination. It swaps segments between the chromosome you got from your mom and the one from your dad, creating new combinations of alleles on each chromosome. This is a massive source of genetic diversity in offspring.
So recombination diversity.
A huge driver of it, yes. And sometimes this process can lead to a curious outcome called gene conversion.
Gene conversion, what's that?
It was first noticed in fungi. Normally, if you cross two parents with different alleles, say allele A and allele A, you expect offspring spores in a 4.4 ratio after meiosis. But occasionally, they'd see weird ratios like 6A spores and 2A spores or vice versa.
So one allele seemed to have been converted into the other.
Precisely. It's as if during recombination, the sequence information from one chromosome was used to overwrite the sequence on its partner.
How does that actually happen? What are the models?
Well, the classic model is the Holiday model. It proposed single-strand nicks, strand invasion, and formation of this X-shaped structure called a Holiday junction that could move along the DNA.
Okay.
But a more current and widely accepted model, especially for initiating recombination, is the double-strand break model. As the name suggests, it starts with one chromosome getting a clean break through both strands.
More drastic than just a nick.
Right. Then enzymes chew back one strand at the break, creating single-stranded tails. One tail then invades the intact homologous chromosome, creating a structure called a D-loop. This invasion allows DNA synthesis to occur, using the intact chromosome as a template to repair the gap caused by the break.
So the repair process itself involves copying from the partner chromosome.
Exactly. This process involves forming Holiday junctions too, which then get resolved. And importantly, these models explain how gene conversion can happen. Two main ways. One, during recombination, you can form regions called heteroduplexes, where one strand is from mom's chromosome and the other from dad's. If they have different alleles, a mismatch, the cell's mismatch repair system might come along and fix it, potentially changing one allele to match the other.
Okay, the mismatch repair system again?
Yep. Or two, in the double-strand break model, if the break happens right within a gene and the chewing back process removes one allele entirely, the subsequent gap repair synthesis, using the homologous chromosome as a template, will naturally copy the allele from that intact chromosome, effectively converting the broken one.
Wow, what an incredible journey today. We've gone from these tiny single-letter typos in DNA.
Right up to these major chromosomal rearrangements and the sophisticated machinery cells used to fix breaks and shuffle genes.
It really underscores that constant tension, doesn't it?
It-
DNA needs to be stable, but life absolutely relies on change, mutation for variation, recombination for diversity, and this whole suite of repair systems holding it all together.
It's a dynamic balance. Understanding it helps make sense of so much inherited disease, evolution, even why we should, wear sunscreen. It's the machinery of life playing out.
Absolutely. It makes you wonder. Think about the sheer resilience built into this system. What other secrets does our genetic repair toolkit hold? What could we learn or even engineer from it in the future?
That's the exciting frontier, isn't it? Constantly uncovering new layers of this intricate process. Well, thank you for joining us on this deep dive today.
Yeah, thanks for listening. And thank you for being part of the Last Minute Lecture family. We'll catch you on the next one.